Welcome to the Chapter 17 video lecture. Chapter 17 deals with organelle evolution and their genomes. There are 5 parts to this chapter. The first part looks at how genes are transmitted through organelles onto offspring, either cells or individuals. Part 2 goes into specialized modes of inheritance depending on the organism. Part 3 discusses mitochondrial interaction with nuclear genes, controlling behavior in terms of metabolic activities. Likewise, Chapter 17 Part 4 looks at the same aspects of chloroplasts. And the last part looks at the theory of endosymbiosis. Once you get over the fact that these organelles, mitochondria and chloroplasts, do carry their own DNA, then one needs to learn how this DNA is copied, inherited, and impacts the rest of the cell's biochemistry. When one talks about organellar inheritance, they're referring to mitochondria and chloroplasts, and not the inheritance of nuclear genes. There is not a single pattern that fits all eukaryotes. However, the most common mechanism is to inherit your mitochondrial and chloroplast genomes and these organelles from one parent, normally the maternal, through fertilized eggs. If this is the case, we call that uniparental inheritance. In a few cases, both parents contribute to the inheritance of these organelles and their genomes, and that's known as biparental organellar inheritance. The situation pertaining to organelles and their genomes is pretty complex because of the sheer numbers involved. One must note that each eukaryotic cell can contain multiple organelles, multiple mitochondria or chloroplasts. And in addition, within each of those, they may contain multiple copies of their chromosomes. This is further complicated by the fact that there's no clear pattern between different species as to the number and size of the organelles and the number of copies of their chromosomes. Neither with the number of genes and the types of genes that are carried on the organelle genomes. An additional level of complication is that the influence, the interaction, the interplay between the genes on the organelle genome and the nuclear genome are different in different creatures. This presents with a lot of information that we have to then learn. Bohr and Correns independently discovered non-Mendelian inheritance patterns in plants in 1908. Bohr was working on geraniums, which are plants, and he noticed that the leaf color was inherited by an odd pattern, and the pattern had to do with the branch upon which the flower was growing. The origin of the pollen had no impact on this coloration. In 1908, scientists were well aware of Mendel's experiments. Bohr took account of this knowledge and began experimentations on geraniums. He came to a surprising conclusion. Whenever he attempted to self-pollinate flowers on a geranium bush, he found that if the bush had different types of leaves on different branches, the flower on which the branch grew would always give rise to the same type of leaves in its progeny. More strangely, when leaves were variegated, that means that they had different patterns on the leaf itself, when flowers of those branches were self-fertilized, they produced not just variegation, but some had white leaves and others had pure green leaves. Figure 17.1 is designed to clarify this relationship. At the very top, we see an example of a geranium bush. So this branch here has white leaves and it has flowers. This branch here has green leaves. It too has flowers of the same color. And the third branch has variegated leaves. If one takes pollen from any one of these flowers or from another bush and introduces it, say, to these flowers, then the seeds that are produced, which are then germinated, will give rise to progeny that all have green flowers. So something about this branch controls the outcome with respect to leaf color. If any pollen is then taken and introduced into these flowers, which are growing on a branch with white leaves, indicated in this row, then all the offspring, once they germinate from the seeds of these flowers, will have white leaves. The situation is a bit more complex when you come to these flowers on these variegated branches. If you take pollen from any plant and you breed it with this flower, the offspring will fall into three categories. Some will be like the parent, they'll be variegated, but others will produce white leaves or green leaves. Bohr went on to make reciprocal crosses between flowers on different branches with different color leaves. In the end, he came to one conclusion, that the phenotype of the branch on which the female parent was growing was key in determining the phenotypes of the progeny. And the overwhelming outcome of the experiments was a logical inference that maternal inheritance was at work in the geranium plants. It wasn't until about 40 years later that other scientists like Chiba and their colleagues that suggested that mitochondria and chloroplasts had their own genomes that were being inherited in certain patterns. Chiba was a cell biologist, and he used the Fulgen stain, known to only stain DNA, to come up with some findings that suggested there was DNA both inside mitochondria and chloroplasts. Let's talk about some terminology that geneticists use in order to refer to the state of an organelle's genomic content. As we alluded, the number of organelles in a cell can vary anywhere from 1 to hundreds and hundreds. And the number of copies of the organelle genome in each organelle can also vary from 1 to many. If in a single-celled organism or a multicellular creature, if all the copies of a particular gene happen to be of the same allele, then we call that homoplasmic, and the organism is said to exhibit homoplasy. If the copies are not the same, then it's heteroplasmic, or the organism is said to exhibit heteroplasy. In this figure, for instance, we look at geraniums. These are plant cells with chloroplasts. If, for instance, all the chloroplasts have all the same allelic copies of that gene, then that's going to be homoplasic. Likewise, if all these chloroplasts contain the same copies of the same gene, then they're going to be homoplasic too. In the last illustration, we have two sets indicating different alleles. Therefore, that is a case of heteroplasy, and the cells are said to be heteroplasmic. With these references, we can then explain what happened in Bohr's work with the geraniums. His conclusion was that the chloroplasts are distributed, segregated amongst the daughter cells in a random fashion. So some go to the left, some go to the right when this cell divides by either mitosis or meiosis. In the case of the green female plant cells, there's only green chloroplasts, so all the progeny will have green. Same thing with the white. The parent, the female parent, only has white non-photosynthetic chloroplasts, and therefore they are inherited by all the offspring. But in the case of the variegated parent female, it depends. If the daughter cell just gets the green ones, then that branch of the future plant will be green. If by chance the daughter cell gets the white ones, then it'll be pure white. And if it gets one of each, or a few of each, then it also, the offspring will be variegated. This is one of the most important findings that students need to learn from this section of this chapter. Turning our attention to the organization of the genome of these organelles, one does not find chromatin. There's no chromatin. There are no histone proteins or the same degree of non-histone protein interaction. But what we do find that there are some proteins interacting with the DNA, and these are very similar to those that we find in prokaryotes, and they also result in the DNA exhibiting the similar organizational relationship that of a nucleoid. However, each nucleoid is not composed of a single genome. It's concatenated with several or many genomes in most cases. Another unremarkable finding was that the genomes of the organelle are independent from the nuclear genome with respect to replication, copying. So an organelle can divide if a cell gets larger, and in order to fulfill that need, the genome of these organelles may divide independently of the nuclear genome. If one combines all the knowledge that exists about organelles and their genomes, we can come to three general conclusions about how genes are transmitted from parents to offspring with respect to organelles. The first is that the growth and division within the cell of the organelles is responsible for dictating how many of them there are, and thus how many will be segregated in which direction when that cell undergoes cytokinesis. Number two, the division and segregation of the nucleoids inside the organelle is also responsible for the genetics. And number three, the replication of the individual organellar genomes within the nucleoids will dictate, as we'll see next. Figure 17.3 illustrates a single chloroplast at the top of the figure, and within that chloroplast we have 5 nucleoids. Each nucleoid has a multitude of genomes. So there's a lot of genomes in this chloroplast. Regardless, suppose there's a mutation in this particular genome as part of this nucleoid. What can happen? Well, when DNA is replicated, this leads to an increase in the number of mutant genomes. Then the nucleoid division takes place. The nucleoids divide from each other, and you can see that here. And they continue to replicate. So now we have a pure nucleoid of mutant DNA. It doesn't have to happen this way, but in this example it has. And then the chloroplast divides in two. Now this mutant nucleoid, there's only one of them. It won't split in half, so it's either going to go to the left or the right. In this example, it's gone to the left. This chloroplast exhibits heteroplasmy, and this one exhibits homoplasmy. If the mutation survives, then there's a probability that in the future some chloroplasts will only have the white mutated genome, and that can give rise to homoplasmic descendants of those cells. Therefore, we can now have heteroplasmic and homoplasmic versions of the chloroplast, 2 homoplasmic and 1 heteroplasmic. Alleles on chloroplasts are segregated through nuclear division. We can use replicative segregation as a term to describe that process. More importantly, it can give rise to mosaic organisms. Organisms such as the example that we saw at the onset in figure 17.1, where a single plant can have different structures in terms of leaf color with respect to the inheritance of chloroplasts. Figure 17.4 tries to illustrate an example of replicative segregation. See if you can follow the sequence of events from the left to the right, leading to these outcomes. In individuals with heteroplasmic arrangements, the interplay between the alleles and genes can lead to what we already know from other chapters, conditions such as penetrance and expressivity, whereby the ratio of mutant to wild-type alleles in all the organelles will vary from cell to cell, tissue to tissue, and even in the population. One can even see more integrative interaction taking place, where the wild-type alleles in one genome may complement the mutant alleles in another genome within the nucleoids of organelles within that cell and within the cells of the organism. Complex. Evidently, for the offspring, the ratio of the number of chloroplast or mitochondrial genomes present in the germ cells will be the great influence leading to the outcomes for that individual. For instance, if your brother is born of heteroplasmic eggs compared to you who may be born of homoplasmic eggs, then the outcome for the two will be different. To complicate matters even further, mitochondria have been observed by time-lapse imagery to fuse together and then to break up again. So fusion and fission are a normal characteristic of mitochondria within long-lived cells, particularly in your immune system. And that can create the potential for individual mitochondria to have genomes of mixed origin. This mixing and breaking it's like mixing dough together. The ingredients will some become homogenized, and that's exactly what happens to the genomes within a population of mitochondria within a single cell. On the contrary, chloroplasts have not been observed to undergo fission or fusion, and therefore their genomes do not undergo any type of homogenization. Let's move on to section 2 that looks at the modes of inheritance of organellar genomes. From a generalized perspective, we can divide the inheritance of organellar genomes into two categories. The first is it biparental or uniparental inheritance? That will determine the outcome. Or, alternatively, the inheritance of these organellar genomes may be independent of the type of parent, and we'll see examples of this coming up. Observations have led to the understanding that different creatures can be grouped together on evolutionary scales. So mammals, they have a very similar form of mitochondrial inheritance, of which there are 4 consequences. And in mammals, it's maternal inheritance that operates. So the father does not contribute, as far as we know, to the offspring's mitochondrial complement. As the mother is the sole source of inheritance in her offspring, we can come to these 4 predictions. The first one says that the mitochondrial mutation inheritance can be based on the mother's genotype. Second, maternal lineage of organisms can be examined. So we can go back in time simply by following the mothers of each generation. And from that we can come to the conclusion that maternal history of all species can be interpreted with respect to the mammals. And human genetic diseases caused by mutational changes to mitochondrial genomes are maternally inherited. This means that all of us have our mother's DNA, irrespective of our sex, and our mothers have their mother's DNA. And this continues backwards. And one of the stories told in an earlier chapter about the Argentinian missing. This is a great example of where that knowledge was used to reunite the kidnapped children with their grandmothers after they were born to captured mothers who were then executed. A pedigree showing a straightforward relationship between mothers and their offspring. So if this mother has a marker or disease that can be followed, then you can see all her children, irrespective of sex, inherit that marker. And her daughters will pass on to all their children the very same marker. The reason you don't see it following in this family or this family here is because the mother in this case is breeding into the family and she's normal, and this mother here, 2-1, is breeding into the family and she's normal. These facts are made use of by genealogy companies or evolutionary biologists to use as a tool, trace back evolutionary relationships. The good thing is there's no recombination of alleles with the father's genome because the father's mitochondrial DNA does not get passed on to the children. We spoke earlier about mitochondrial genes having alleles. Can these alleles result in disease? And the answer is absolutely. So mitochondrial mutations can result in human genetic diseases, and in fact any disease of any creature that harbors mitochondria. That will be all eukaryotes with 1 or 2 specific exceptions. These mutations tend to be highly pleiotropic because they have to do, in most cases, with biochemical pathways involving oxidative phosphorylation. If you have forgotten what the word pleiotropic means, this is a great time to pause the video and go and look it up on the internet. One example of a famous disease to do with mitochondria is Leber's hereditary optic neuropathy, which has a pleiotropic effect including heart abnormalities. Leber's is one of many diseases that have now been mapped to the mitochondrial genome in humans. You don't need to learn any of these, except if you look carefully, you will see that the magnitude of phenotypic effects is huge. situations become very complex because in mitochondrial inheritance, penetrance becomes a big factor. Again, if you don't know what penetrance means, this is a great time to go and look up after stopping this video. Any affected child will have an affected mother, but the opposite may not be true. An affected mother may not have all her children affected to the same degree, and this is because of incomplete penetrance, due to a combination of factors, including heteroplasmy, the influence of nuclear genes, and environmental factors. Indeed, if a human mother is heteroplasmic, she may produce eggs that are homoplasmic or heteroplasmic, giving rise to both types of offspring. Then pedigrees of this type become apparent. Please note there is a small error in this figure. They have misnumbered row number 2. So this should be 1, this should be 2, this should be 3, 4, 5, 6, 7, 8. So apologies there. Regardless, the effect is still apparent. This mother is affected with LHON, and she should have given it to this son and this daughter, as well as this daughter, this daughter, this daughter, and that daughter. But you can see the pattern illustrates otherwise. Therefore, this is a case of incomplete penetrance. Cell biology of human mitochondria is interesting in itself. When human oocytes are first looked at, they do contain a few large mitochondria, about 10. But these will then go on to divide multiple times by fission into smaller mitochondria, and their numbers may increase up to 2,000 genomes. Interestingly, the origin of this 2,000 mitochondria, being these 10 or so large mitochondria, increases the possibility of producing homoplasmic offspring. Figure 17.7 is a compound figure which brings together all these facts. So looking at panel A, we have homoplasmic segregation. This mother is 100% mutant, and her children are 100% mutant, every single one of them. In the lower pedigrees, the mother is heteroplasmic, and depending on the way the mitochondria segregate in the formation of the oocytes, some of her children may end up with 100% down to 0% in terms of the mutant phenotype. In panel B, we see the premodal germ cell contains both wild type and mutant mitochondria, where the proportion of the blue mutant ones is lower than the red wild type. Then during egg production, we see that there's a random segregation of mitochondria amongst all the primary oocytes, which then leads to a different ratio in each one. So in the bottom primary oocyte, we have far more mutant mitochondria than we do wild type. And when these go on to mature oocytes, they will replicate and the proportions stay roughly the same, and there's a high incidence of affected outcomes. If this is fertilized by a sperm, the individual will have a high penetrance of this disease compared to this oocyte if that's used for forming the offspring. As with the X chromosome in females, mitochondria can do something very similar. And when they do it through replicative segregation, heteroplasmic individuals may end up with different tissues in their bodies that contain different proportions of wild type to mutant mitochondria. As cells divide by mitosis, the positioning of the mutant versus the wild type mitochondria on either side of the cytoplasmic furrow as the cell divides will determine the outcome for that lineage of cell. The biology of Saccharomyces cerevisiae, which is a yeast, is very important so that we can understand a section later on. This single-celled yeast can grow both aerobically in the presence of oxygen and anaerobically in the absence of oxygen. In either case, it continues to grow. However, it grows more slowly in anaerobic conditions compared to aerobic conditions, simply because of the amount of ATP produced. The mitochondria are important in aerobic conditions because they become activated and the presence of oxygen drives the formation of water and therefore the use of oxygen. It was in the mid-1950s that the Petite mutations were discovered by the work of Efferusi, whereby he noticed that on some fermentative media, The colonies were growing smaller than the normal wild-type colonies, so he called the large normal colonies grande and the small colonies petite. These small colonies, the petite colonies, when transferred to media that only permitted aerobic growth, that killed them all. Upon further investigation, the petite colonies fell into three distinct classes. The first class relied on segregation of nuclear genes to control their phenotype. So these were known as segregational petites or nuclear petites. These nuclear petites demonstrated that nuclear genes are also needed for full mitochondrial function to take place. The remaining two classes had nothing to do with nuclear inheritance. In fact, their patterns of inheritance suggested mitochondrial genome inheritance. And those were the neutral petites and the suppressive petites. Figure 17.9 re-illustrates this. That if there's a nuclear mutation, it follows Mendelian genetics. Where there's a mitochondrial mutation, it does not. One thing we have to understand is that during yeast reproduction, during the process of forming the zygotes, we get something called a tetrad. A tetrad is a cell that contains the four products of meiosis still held together in the same cytoplasm. So please remember that as we move forward to discuss this analysis. Let's examine the biology and genetics of the natural petites. So when natural petites are crossed to wild-type cells, the diploid zygote grows normally, and these tetrads are formed inside, and they contain only wild-type spores. Figure 17.9c shows this cross. A wild-type cell producing a grande colony with a neutral petite. When crossed together, the cells, they always produce colonies that are wild-type. The reason for this is quite simple. The neutral petites contain no mitochondrial DNA whatsoever, and so their offspring all have mitochondria derived from the wild-type parent. The same experiments performed with suppressive petites results in a different outcome. Once again, you take your wild-type and your suppressive petite, you cross them together, and all the progeny are petite. There are no wild types. Biochemical examination of the mitochondrial genomes of these petite colonies reveals that they all contain some type of deletion in portions of their mitochondrial DNA, which when sustained for future generations, they tend to become worse and worse by developing larger scale rearrangements and duplications that lead to eventual loss of mitochondrial function. Then the questions asked are, what's happening? What's going on? Do we understand? And the answer is not really, but there are two possibilities. The first is that the suppressive petites, their mitochondria may replicate faster than the wild type. Maybe they possess multiple replication origins, therefore ensuring that they outcompete the wild type versions. The second possibility is that the genomes of the two types of mitochondria may fuse, giving rise to genomic rearrangements that induce rearrangements of the wild type mitochondrial DNA also. So like an infectious agent. In summary, we have various types of inheritance when it comes to mitochondrial genomes. The next section of the chapter looks at mitochondrial genetics and their role in energy supply for the eukaryotic cell. The Krebs cycle and the electron transport chain, the oxidative phosphorylation proportion of ATP generation, are all taking place inside the membranes of mitochondria. In many species, mitochondria participate in other processes in addition to just ATP production. It turns out that the mitochondria, its genome, is incapable of supporting itself. Many of its functions have been deferred over to the nucleus. So the nucleus is responsible for making proteins, which are then exported from the cytosol into the mitochondria, where they are able to perform their duties. A strange relationship. Another thing is that in different species, this relationship varies. So in some cases, some genes are in the mitochondria. In other species, those same genes are in the nucleus. and the number of genes in some species are greater in the mitochondria than in the nucleus. The important thing is that ribosomal RNA, for making the ribosomes that are contained within the mitochondria, those genes are always made inside the genome of the mitochondria. The genomes of mitochondria are normally circular, just like bacteria, but there's growing evidence that they can change forms and become linear. Indeed, in some species, they are only found in the linear form. In most cases, the mitochondrial genome is a single molecule, normally circular. But in a few species, these genomes have been fragmented into multiple chromosomes. Figure 17.11 shows a relationship between different species. So here we have Tetrahyma. It has a linear mitochondrial DNA. A human is a single circular DNA in most cases. Then we have other species like an amoeboid, an amoeba. It has multiple chromosomes. And then we have other types of fungi that have multiple circular chromosomes. As we stated at the beginning of this video, the mitochondrial DNA are not associated or packaged with histones into chromatin. Instead, they are anchored through other proteins to the inner mitochondrial membrane. Although today the gene content and size of these mitochondrial genomes do vary a lot between eukaryotes, all the evidence points to a hypothesis that states that all mitochondrial genomes are descended from a common bacterial ancestral genome, which had thousands of genes, and the present-day situation reflects differential gene loss by different species. Figure 17.12a shows a human mitochondrial DNA map compared to a protozoa that has much more. If you were asked in the future by somebody, how big is the human mitochondrial genome, you would say it's 16,569 base pairs. That places us about average for different eukaryotes. Figure 17.12b shows the relationship between the size of the mitochondrial genome and the classes of genes that exist. The DNA of mitochondrial genomes is transcribed in place by a RNA polymerase, a single RNA polymerase, which is similar to the RNA polymerase found in bacteria. The origin of this polymerase can vary. In some species, it's made in the mitochondria itself by its own ribosomes, and in other species, it's made from a nuclear gene and then imported. The organization of these genes and their transcription regulation mechanisms seem to be more aligned with bacterial operons than they are with the eukaryotes. In mammals, at this point, just 3 polycistronic messenger RNA transcripts are made from 3 promoters in mitochondrial genomes. In other eukaryotes, this number is different. The ribosomes that operate inside mitochondria, they resemble the structure and sizes of ribosomes from bacteria. All the ribosomal RNAs that are structurally used to make the ribosomes of mitochondria, they are encoded on the genomes of mitochondria. But the proteins, they may come either from mitochondrial genes or from nuclear genes. In some rare cases, in some creatures, there are Shine-Delgado sequences which drive protein synthesis, but for most, these do not exist. In most species, the genetic code used by the mitochondria is the same as nuclear genes, with some minor modifications as listed in Table 17.1. UGA is a stop codon on the nuclear DNA. It is also a stop codon on land plants, but it codes for tryptophan in all these other branches of the eukaryotic tree of life, including us. In addition, in mammalian mitochondria, the third base wobble is more flexible compared to nuclear genes. And in some species, the mitochondrial transfer RNAs are coded by nuclear genes and then imported from the cytosol into mitochondria. All pretty strange. Let's move on to section 17.4. And now let's pay attention to chloroplasts. The chloroplasts are a specialized form of plastid. These plastids come and develop into many structures, including those that hold color in petals or store starch as in potatoes. Cells which interact with light normally have some of their plastids converted into chloroplasts. Chloroplasts have three membranes, an outer membrane, an inner membrane, and a third membrane called a thylakoid membrane. The thylakoid membrane is responsible for demarcating the space between the lumen of the thylakoid and the stroma of the chloroplast. Both these spaces can store protons to different degrees, therefore driving the production of ATP. The thylakoid membrane itself has many proteins embedded in it that are important in photosynthesis, most of which are encoded by nuclear genes and thus have to be manufactured in the cytosol and then imported into this organelle. Figure 1714 shows a typical chloroplast and then a cartoon showing the arrangement of the thylakoids inside the stroma of the chloroplast. The thylakoid membrane itself is responsible for a multitude of protein systems, including the ATP synthase and photosystem 1 and photosystem 2, as well as for other proteins. Once again, many structural features of chloroplasts resemble those of bacteria, and because bacteria and mitochondria resemble each other, all three are related. The genomes within chloroplasts are generally circular, although some linear cases are known, and these genomes are less diverse than mitochondrial genomes. And once again, the numbers of genes and the exact type of gene varies amongst species. Most of the genes carried on the chloroplast genomes have to do with photosynthesis, whether they come from plants such as lettuce or the liverwort, which is a type of moss. Everything on this slide with a purple background suggests similarity to bacteria. So transcription and translation processes within chloroplasts, similar to bacteria. The RNA polymerase, similar to bacteria. The parts of the DNA that it recognizes as promoters, similar to bacteria, the -10 and the -35 consensus sequences. The transcripts are not capped, similar to bacteria, nor are they polyadenylated, similar to bacteria. But in some cases, introns are removed from messenger RNAs of a few genes. So this is unusual. They do have introns, but in some genes, not all genes. The ribosomes themselves are similar to those of bacteria in size and construct. So there are no deviations from the universal genetic code as seen in mitochondria. RNA editing is a term applied to RNA once it's been transcribed, where its sequence is thereafter changed before it goes on to become used by the cell. So RNA editing has been observed both in mitochondria and in chloroplasts of land plants. So land plants utilize RNA editing to a greater degree than other organisms. And the editing of organelle RNAs in plants does not utilize a guide RNA, as it does in animal cells. The messenger RNAs of several genes encoding proteins involving photosynthesis are edited. Interestingly, it appears that RNA editing, which uses both RNA and proteins, those proteins are made in the nucleus and then transported through the transcription translation in the cytosol into the mitochondria. And we have lots of examples like the CCR4 gene in Arabidopsis that results in mutations in the nucleus that can cause chloroplasts to be deficient in photosynthesis. An example of RNA editing is demonstrated by figure 17.16. The last part of the chapter addresses the endosymbiosis theory and explains in the context of mitochondria and chloroplasts their evolution. The symbiosis part of the word endosymbiosis refers to a mutually beneficial relationship between biological organisms. Endo simply means the presence of one inside another. So endosymbiosis has to do with this relationship between mitochondria, chloroplasts, and the cells in which they reside. So a lot of evidence is now suggesting that mitochondria and chloroplasts were free-living bacteria at one time, and that they took part in ancient infections of eukaryotic cells. Due to mutation or error or some other unknown event, these invaders established symbiotic relationships with their hosts and have led to the production of these organelles. Thus the endosymbiosis theory. We've already mentioned a lot of evidence in the previous two sections, but here we have a concatenation of the evidence. First thing is the double membrane system of chloroplast and mitochondria, where the outer membrane resembles current-day eukaryotic cells, and the inner membranes resemble the membranes found on bacteria. The size of the organelles is similar to those of modern-day bacteria. The nature of the DNA packaging is similar to that of bacteria and completely different to nuclear DNA. The transcription, translation, and machinery used by the organelles closely resembles in size and structure that of bacteria, and the protein coding sequences of organellar genes are more like those of bacteria than they are of eukaryotes. Evidence at this time suggests that mitochondria are monophyletic. That means they all descended from a single ancestor. This points to a single endosymbiotic event that gave rise to the mitochondria. Following a lot of biochemical investigation, one hypothesis suggests that an anaerobic cell living in the past acquired an aerobic endosymbiont bacteria, and that endosymbiont was a free-living alpha proteobacteria. Equally, further research indicates that chloroplasts are also monophyletic. That means they also descended from a single endosymbiotic event about 1.2 billion years ago. The closest living relatives of chloroplasts are free-living cyanobacteria. Comparison of the genomes of these cyanobacteria with the chloroplasts suggests that the chloroplasts contain far fewer genes than do the cyanobacteria. So there was a large-scale loss of genes that has taken place during that time period. Figure 1717 does a marvelous job of trying to relay the relationship between the branches of the tree of life and the movement of these organelles or bacteria through endosymbiosis. Hang on a second. Phylogenetic studies suggest that multiple secondary symbiosis has taken place, in which some eukaryotes acquired a photosynthetic eukaryotic symbiont. So what does that mean? That means if you follow one of these traces, this one in particular, you can see that first mitochondria were established, and then there was a second endosymbiotic event that led to two internal plastids inside the eukaryotic cell. Further, there was a horizontal transfer into some lineages of already endosymbiotic eukaryotes. As mentioned before, many of the genes from chloroplasts and mitochondria have been lost and relocated to the nuclear genome in these endosymbionts. Indeed, there's evidence that the transfer of genes from the organelles genomes to the nuclear genomes is currently taking place. So it's not just an ancient thing, it's a more recent transfer. This evidence comes from comparative genomics, where the genomes of mitochondria are compared with nuclear genomes of eukaryotes and bacterial genomes. Analysis of the sequences that are highly similar must have taken place more recently because mutations have not accumulated to the same degree as in other regions. Figure 17.18 shows the shuffling of genes between the endosymbionts and the organelles. The thickness of the arrows is a clue as to the degree of migration of different genes. One such dataset is shown here. Arabidopsis, a weed, and 3 cyanobacterial species have been compared by computers, and the data shows that about 4,300 nuclear genes in Arabidopsis have a cyanobacterial origin. Likewise, comparisons between eukaryotic nuclear genes and alpha protobacteria has detected 630 nuclear genes that are derived from an endosymbiont that gave rise to these mitochondria. Figure 17.19 shows the migration of genes between the endosymbiont that we think was involved in the formation of the chloroplast and the nucleus and mitochondria of present-day Arabidopsis. The numbers refer to the migration of proteins. Mutational analysis has shown that the transfer continues until more recent times. So with respect to nuclear mitochondrial sequences, known as NUMTS, they are genes in the nucleus that are recently derived from mitochondrial genomes. With nuclear plastid sequences, NUPTS, they are genes in the nucleus that are recently derived from the plastid genomes. But this transfer is not always one way. Sometimes you have genes going from the nucleus out to the plastids, or the mitochondria. And this rate is very high, and we're not sure why at this time. As with chromosomal translocations, when genes move from one genome to another, they have to be transcribed at the correct rate. Therefore, they must acquire the appropriate expression in the nucleus that allows them to then continue to function for the benefit of that cell. This may require appropriate enhancers and promoters, as well as silencers. In addition, once the gene has translocated to the nucleus, then the proteins that it makes have to then gain entry back into the organelle in order to function, and that requires additional processing. One of these is the amino acid terminal signal sequence that is required to gain entry back into the organelle. So that has to evolve correctly as well. These extra pieces of polypeptide are known as signal sequences. Normally between 15 and 25 amino acids. They're normally at their amino ends of the amino acid chain. Different sequences, different amino acids combinations, target different proteins to different destinations within the cell, including different locations within that organelle. Recall that the mitochondria has 2 membranes, therefore there are 3 compartments: the cytosol, the intermembrane space, and the matrix, and the chloroplast has 4 spaces because it has 3 membranes. There's also another level of weirdness. So we now have discovered that in the past, certain genes resided in a certain organelle. Those genes may have moved to the nucleus, but their proteins are no longer targeted to that original organelle, but the proteins are sent to another organelle. For example, in the case of Arabidopsis, less than half of the genes originally on the endosymbiont cyanobacterium are now targeted to the chloroplast. Conversely, a number of proteins now that do end up in the chloroplast did not originally originate from the endosymbiont that was the cyanobacterium. In reality then, if we assume certain privileges, we can say that if the original cell was a bacterium, the endosymbiont, then The flow of genetic information from the genome of the endosymbiont to our genomes could be represented in this fashion here. We could say that there's extensive gene flow from bacterial endosymbionts to our nucleus. That's resulted in a significant number of our genes being of bacterial origin. But those do represent a small fraction of the total nuclear genome that we possess. What is the origin of the majority of the genes then, if they're not bacterial? So scientists have come up with two models to propose this. In the first model, the original host cell was already a eukaryotic cell that had a nucleus, but it didn't have a mitochondria, and subsequently acquired the alpha proteobacterium as a symbiont. So the eukaryotic cell existed already. In a competing model, the original host cell was not yet a eukaryotic cell because it didn't have itself a nucleus. It was simply a bigger bacterium that acquired the endosymbiont. And subsequently, eukaryotic features developed inside this larger bacterium, such as a nuclear membrane. This hodgepodge of activity simply complicates the analysis of what happened in the past. Once we thought that there were eukaryotes that didn't have any mitochondria, such as Giardia. But now, with better tools, we're able to detect what looked like mitochondria inside these cells. So the mitochondria of Giardia have been reduced to small membrane-bound structures that themselves lack their own genome. And these are called mitosomes. So taking the first model at good faith, that suggests that at one time, eukaryotic cells have existed that had no mitochondria. Could they still exist today? There's been no evidence now that this case has been overturned. How about the second model? The second model suggests that there should be no intermediate eukaryotes that lacked mitochondria. And that appears to be the case. Much of this evidence has come from the examination of yeast cells, Saccharomyces cerevisiae, and comparing that sequence using computer programs with 2 bacterial species and 1 archaeal species shows that the majority of the yeast genes in the nucleus can be divided into 2 sets: informational genes and operational genes. Informational genes, think about it, they interact with components of the cell that carry information. So that would be DNA and RNA. So informational genes encode proteins that perform processes such as DNA replication, chromosome packaging, transcription, and translation. And from the case of the yeast, these genes resemble those of the archaea, Methanococcus. Operational genes are genes that are part of metabolic pathways. And most of these resemble bacterial genes, in the case of the Saccharomyces cerevisiae. We need to remember that present-day cells may not actually resemble the cells which existed at the time these endosymbiotic events took place. So according to some authors, the putative host may not be in existence today for us to compare our genomes with. So present-day archaeal cells have evolved themselves from their ancestors. So if one of them was the ancestor of eukaryotic evolution, it doesn't exist any longer. Figure 1720 shows one of many hypotheses for the evolution of eukaryotic cells. And it says that cyanobacteria were important at a certain point, and they gave rise to modern-day plants. Likewise, proteobacteria may have been the origins of mitochondria. And the nuclear DNA may have come from an archaea. With continued endosymbiotic events, secondary and tertiary, etc., whenever one cell acquires another cell inside it, the genes of the endosymbiont, one inside, its genome transfers to the outer cell, the host cell. And if that host cell becomes an endosymbiont in another bigger cell, then its genome will begin to transfer to the genome of its host. The term nucleomorph is applied to the genome of the endosymbiont. These migrations, horizontal and lateral between generations, is all key in giving us who we are today. So if you're looking for the meaning of life, the meaning of life is pretty interesting. It's intertwined between migrations of endosymbionts, with nuclear migrations, with migrations of proteins from nucleus back into the organelles. So there's no direct, clear case that we can follow in our history. We are a result of convoluted migrations of genomes and biochemistries. Strange examples of these migrations exist all around us, and there's many more to be uncovered. So the story is ongoing. Thank you.